When a modern DNA sequencer ignites, it doesn't quietly hand you an answer. It blasts you with billions of chemical signals, translating human biology into a blinding digital storm of A's, C's, T's, and G's. But standing alone, that torrential downpour of text is completely unreadable. It is chaotic, unanchored, and stripped of any biological context. The scale is staggering. To find a single genetic mutation responsible for a rare disease, a researcher has to sift through 3 billion normal letters without losing their place. Here is a multi-tiered funnel designed to relentlessly filter that raw sequencing data. It takes the chaos at the top, forces it through strict computational checkpoints, and squeezes out a single actionable data point at the bottom. To find the signal in the noise, every read must survive 3 mandatory formatting checkpoints: FASTQ, BAM, and VCF. The first stop in this pipeline is the FASTQ format. Think of it as the raw catch hauled straight from the sequencing machine, untouched and unmapped. This chromatogram shows exactly how the machine makes that raw catch. Optical sensors record chemical flashes as colored peaks, which the software then translates directly into the digital letters you see below. To organize this output, a FASTQ file packages every single read into 4 strict lines. Line 2 is the actual biology. These are the raw A, C, T, and G base pairs caught by the sequencer's camera. But the hardware isn't perfect. That's why line 4 contains the quality string. Each keyboard symbol here maps 1-to-1 directly beneath a specific DNA letter from line 2. These symbols represent a Fred quality score. If a letter gets a score of Q30, it means the machine is 99.9% confident it identified that specific base pair correctly. We now have millions of high-confidence reads, but they exist in a vacuum. A FASTQ file provides the letters, but it cannot tell us which gene or chromosome they belong to. To make sense of those reads, we take our unknown sequences and map them against a standard, known reference genome. This alignment block illustrates that process. You can see the vertical lines locking the top sequence into the reference below it, while the blank spaces reveal the gaps where they failed to align perfectly. The results of this mapping are stored in a SAM file, a human-readable text document that records the exact chromosomal coordinates where each read finally snapped into place. However, human-readable text takes up too much space. To process massive datasets, the computer compresses the SAM into its binary twin, the BAM file, which works alongside an index file so the machine can instantly search millions of coordinates. Inside these files, the precise nature of every alignment is described using a highly specific grammar known as the cigar string. It reads like a cheat code for biology. M tells us a base matches the reference, I indicates an insertion of new material, and D flags a deletion where DNA is missing. At this stage, our data is perfectly organized by geographic location. But a BAM file still carries the entire weight of the genome, including the millions of identical, healthy DNA sequences we don't care about. The final computational hurdle requires stripping away the 99% of the read that perfectly matches the reference genome. We pour the remaining data into the variant call format, or VCF. This container is designed exclusively to hold the genetic anomalies left behind. This VCF row breaks down exactly how a mutation is recorded. The data is separated into strict vertical columns, marking the position on the chromosome alongside the specific biological change. By comparing the ref column, what the standard human reference genome expects to see, against the alt column, we isolate the exact alternate letter found in our patient. It doesn't stop at the letters. The QAL, filter, and info columns attach critical metadata to that anomaly, proving mathematically that this mutation is a biological reality and not a glitch in the sequencer's camera. This single file represents the end of the line. We've thrown away billions of useless data points to isolate pure biological insight. This workflow visualizes the entire data lifecycle. We capture the raw reads and their quality scores in FASTQ. We map them to a geographic scaffold using BAM. Finally, we distill the chaos down to pinpoint mutations in VCF. This pipeline functions as the global standard for genomic exchange, allowing different labs and machines to speak the same digital language. This mechanism lets researchers spot a tumor's precise driver, allowing a targeted molecule to lock into a mutated cancer cell. By forcing noise through FASTQ, BAM, and VCF, We translate raw data into actionable